So good evening everyone. We're going to go live in a couple of minutes time and we're going to go through additional B2 biology content. Right, good evening everyone. I think we'll make a start. This is the Revision Monkey Hangout for additional science and we're looking at the biology aspects tonight. And like we've done for the chemistry and the physics, we are going to try and compress the entire course into an hour session of the key content. Obviously the caveat to all of this is we're only going to be able to go through the absolutely main aspects of the course. You are going to need to ensure that using the precise learning, you've checked off all the key points that are going to come through into the exam. But obviously we'll go through all the key parts. So starting through, here are the main topics that we're going to look at tonight. We've got the cells part, which is for structures, specialization, organization, and the processes that we find at a subcellular level. We've got plants, their structure and photosynthesis, and around that, things like rates of photosynthesis. We've got organism distribution, how we can count the number of animals, transects, etc. A little bit about enzymes and some specifics that they tend to ask in the exam. And then the second half that we've been teaching will then focus in on DNA, the structure, processes such as mitosis for everyone, and if you're doing higher, meiosis. A little talk about stem cells, and we'll look at some genetics. So the founder of genetics, Mendel, given disorders, and how we can do genetic crosses to find out ratios and percentage chances of inheriting genetic disorders. And then finally, we're going to end on life on Earth aspects, which include fossils, extinction, and speciation. So let's make a start. Well, the first kind of key concept we need to be aware of are cells and cell structures. And again, we're taking some pictures from the internet here to resemble some classic cells. And obviously in the exam, maybe drawings very similar to this. Some key parts that we need to be aware of in the exam that they tend to ask us. And we're just going to go through them now. So the first one that we're going to find is the nucleus, which is in an animal cell and a plant cell over here on the right hand side. And I'll just clarify with you, I'm going to label all the animal cells to begin with over here on this left hand side, and then we'll look at the plant cells and we'll end on yeast and bacteria. So specifically the nucleus, that's a likely cell organelle to come up, and this is containing the genetic information it's controlling the cell. The second one, which is less likely to come up, is the cytoplasm, but just be aware that it's the jelly-like substance. It's where the chemical reactions take place, where we find lots of the enzymes. We've got ourselves a cell membrane. Now the cell membrane is there to essentially allow certain substances in and out of the cell, and it holds the cell together. And then the new year 11 parts, we've got these organelles called mitochondria. Now mitochondria, again, quite likely to come up if they're going to ask this, is the power plant of the cell. It's where respiration takes place. And then we've got ribosomes at the bottom. And the ribosomes are where proteins are produced. Now a plant cell has all the following that the animal cell had, so I won't relabel them, but it then does have some very specific extra parts. It's got a cell wall, and the cell wall is there to add a bit of rigidity to the cell, gives it a bit of support, a bit of strength. We've got a vacuole, which is a storage part of the plant. Contains cell sap, sugars, salts, things like that. And finally, chloroplasts, which contain chlorophyll, and it's where photosynthesis occurs. Now, if I was going to be predicting, I'm probably looking at things such as the nucleus, mitochondria, ribosomes, and chloroplasts. They'd be the most likely things to come up. Again, like I said at the start, it's not to say any of these could come up, but they're the likely ones. Outside of that, there are two specific other variants of cells that we just need to know for the exam. We have ourselves a yeast cell. Now, yeast are a strange type of microorganism. They do have a nucleus, and they do have a cytoplasm, and they also have a cell membrane. So very similar to an animal cell. But a difference is they also contain a cell wall. Finally, we've got our simple bacteria. And again, some similar parts. We've got ourselves the cell membrane. We've got our cytoplasm. We've got some genetic information, some genetic material here. You'll notice it's not in the nucleus. So it's loose genetic material, and they also have a cell wall. So a couple of differences with our bacteria and our yeast. Again, for me, the key bit is for bacteria, the genetic material is not within a cell organelle per se, it's not within a nucleus. And for yeast, they have everything an animal has, but they also have a cell wall. So that's our first key part, looking at the types of cells, and they may well ask a couple of questions around this. Now, moving on, we're going to look at some cell specialization. So how some actual specific cells are specifically adapted to function in the way that they do. So I'm going to start bottom right with an animal cell over here. Very specifically, we've got ourselves a red blood cell. Now, a red blood cell is essentially a shape which has a very large surface area to allow it to carry lots of oxygen. And actually, in part of that, it doesn't have a nucleus when it's mature. And that just gives it a little bit more space to carry that oxygen. Now we've got hemoglobin in there as well, a pigment to allow that, but that's for key specialization for it. If we look at some gametes, a sperm and an egg, well, a few specific adaptations for each. The sperm has a tail, allowing it to swim. It's got lots of mitochondria in it, and the mitochondria give it lots of energy, so it's able to move where it needs to move to. The egg, on the other hand, in comparison to the cell of a sperm, is much, much bigger. And this is because actually it's got a very large cytoplasm. And the cytoplasm is essentially an energy store. Now, finally, if we look at a plant specialization, there are two key parts that we're going to look at. The first are our leaf cells, our palisade leaf cells. Now, if we think what a leaf is there to do, its key role is for photosynthesis. So it wants a mechanism to get as much carbon dioxide, or water, and then finally light as possible. So specifically with the leaf, it's all about absorbing light. And to that end, it's got a very large number of chloroplasts. Counter that to, say, a root cell, which is not adapted for photosynthesis, then you'll find that no chloroplasts are deep within because no light will be being absorbed. Outside of that, some very specific cells within a leaf cell known as guard cells. And the guard cells are to the left and right hand side of very specific pores known as stomata. And so to aid photosynthesis, we need a process by which carbon dioxide can enter the leaf cell. And it can do that through the stomata, through the hole. But a problem with that, which we'll talk about later, is potentially the loss of water. And so these guard cells are found either side of that stomata pore, and they can go flaccid or turgid with water, depending on what the plant needs, and that will open and close the stomata. And so that will either allow the pore to be open and therefore carbon dioxide come in at potentially the expense of water, and vice versa, it can close down, reducing any water loss, but also reducing the ability for the plant to take in carbon dioxide. So here are some key specializations that might again come up in the exam. Specifically, they might ask us to start looking at cell organization, in which case we might need to look at drawing a leaf cell. And we'll come back to the actual drawing, the schematic of that later on. Well, let's talk about cell organization, and let's start with an animal. The specific one that we're going to look at here will be the digestive system in an animal. Before we get there though, let's go through the basics. The simplest part of our cell organization is the cell. And the cell is our building block. And as we've seen in the previous slide, animal cells and plant cells are quite different. Now, a group of similar cells can work together, and they're known as a tissue. And they're going to carry out a particular function. The specific examples that we're going to need to know in the exam are things such as muscular tissue, so allowing in the example of our digestive system within the stomach to churn up the food. We need to be aware of glandular tissues. So that is allowing us, again, in our example of a digestive system, to secrete enzymes. And then we've got the epithelial tissues. and they cover parts of the body. So things like the gut has a covering of epithelial cells. So these simple cells are grouped together to form a tissue, and then we can find that organs are tissues that are organised collectively. So it's different tissues working for a given purpose. So for example, the stomach is an organ made up of those different tissues that we've just mentioned above: the muscular, the glandular, and the epithelial. Finally, the organs can work together to form an organ system. And again, if we go back through our digestive system, we're talking about things like the stomach, the small intestine, the large intestine, the liver. So organisation is cell to tissue to organs to organ systems. And there are two that we need to know for the exam. The first is to do with animal cells, specifically within the digestive system. And the second is to do with plants, looking specifically at a leaf cell. So if we take our digestive system, we go from an epithelial cell to the tissue into the stomach, and the stomach does a variety of processes, then passing down through and working together with the pancreas, the liver, the small intestine, and the large intestine to essentially digest and absorb food. Now, the key parts for all of this kind of aspect I do apologise, it would appear that the pen isn't currently working. So if you just give me 10 seconds, I'll see if I can get my pen working. Hello everyone, just to let you know, we're having a slight technical issue here with my pen, so I'm going to start the presentation again. I'll be back on air in under 5 minutes just to go through the key part. So bear with me on this one, I'm afraid. Right, good evening everyone. I'm going to try that session again. I'm really sorry about that earlier. It would appear that my pen wasn't working on the additional biology hangout. So I'm going to start again. I'm really sorry if you tuned in previously to that, and if you were listening to me, I'm very sorry you're going to have to listen to me again, or you can go and have a cup of tea and skip back and come back in 5 minutes time as I go through the first part. So hopefully this session will now work without the gremlins, and let's make a start. So as previously said before, we're going to have a quick look at the key topics as an overview for the Biology 2 Hangout, and that's going to include cells, which includes specifically the structures, the specialization, the organization, and the processes. We're going to look at the plants, and with that, the structure and the photosynthesis aspects. We're going to look at some organism distribution, and that's to do with quadrats and transects. Then we're going to go into enzymes, catalysts, digestion, respiration, and how they're used in industry. And then the second half, we're going to look at DNA, which is structure, mitosis, meiosis, and stem cells. And we're going to go into then finally genetics, looking at Mendel, the disorders, and the diagrams within. And we're going to end on the life on Earth part, which is looking at fossils, extinction, and speciation. So hopefully this time without too much of a hitch, we'll move on and we'll start with cells. So looking at this specifically, in the exam, we're going to find ourselves looking at two specific parts to the questions. We're going to be hopefully being able to identify specifics to do with animal cells. And then after that, we need to move on and look at plants. So let's take a start here. Look at the key organelles that we find ourselves with an animal cell. The first part is the nucleus. Now the nucleus is where our genetic information is contained, and it's controlling the cell. We then have the cytoplasm. We find with the cytoplasm, this is where chemical reactions take place. It's where we find the vast majority of the enzymes. We have a cell membrane. The cell membrane allows certain substances in and out of the cell. It's the jelly-like substance. We then have mitochondria. And with mitochondria, this is where we find respiration taking place, the release of the energy for the cell. And finally, we have our ribosomes. And ribosomes are where our proteins are synthesized, where they're made. Now, with a plant cell, we have exactly the same organelles as we find in an animal cell. So I won't relist them, but there are a couple of very specific extra ones that we have. We've got a cell wall. The cell wall offers some rigidity to the actual cell of the plant, some support. We have a vacuole, which acts as a storage of sugars and salts. And then we have chloroplasts. And chloroplasts are our organelles which allow for photosynthesis. They contain the chlorophyll and they're key in the photosynthetic process. Now, the key ones out of all of these, and again, as we said previously, there's a huge amount of content in the biology, and we're doing a skim today of the key aspects. So they could, of course, ask any of these in the exam. But I think the key ones that we should be focusing on are probably the nucleus, controlling the cell, the genetic information, mitochondria, where respiration takes place, ribosomes, where the proteins are synthesized, and then from a plant, chloroplasts, for where photosynthesis takes place. Now, at this point, there are two other types of cell that we need to be aware of. These are yeast and bacteria. And they're similar in certain respects to animals and plant cells, but they've got some very specific parts to them that we just need to be aware of. So if we look at a yeast cell has a nucleus, it has a cytoplasm, and it also has a cell membrane. So it's just like our animal cell. But it does have one key difference. They possess a cell wall, which is clearly very different to our animal cell. And then finally, with bacteria, again, they have a cytoplasm, they have a cell membrane, they have a cell wall. But the key difference here is they have no nucleus. Their genetic material is just found loosely within the cell. So animal cell, nucleus for control, genetic information, mitochondria for respiration, and ribosomes for protein synthesis. Plant cells, all of the above, along with chloroplasts for photosynthesis. Yeast cells, very similar to an animal cell, but they have a cell wall. And bacteria, a bit of a mix of both, cell wall, cell membrane, cytoplasm, but genetic material that isn't within a nucleus. So moving on to our second part is now looking at how certain cells are specifically specialized, how they are adapted to function for the exact need that they're there to serve. So a couple of examples here that we need to be aware of. First is quite a simple one. We're looking at a red blood cell. Now our red blood cell, when mature, has a very large surface area. And it's because its main role is to carry oxygen with a special pigment called hemoglobin. So having a large surface area means lots of oxygen can be carried. And to aid that, when they're mature, they actually lose their nucleus. The nucleus goes. And so that allows even more space for oxygen to fill. If we're looking at our gametes for sperm and egg, two specific parts really. First of all, the sperm has a tail for mobility to swim, and it's got lots of mitochondria. And if we recall, mitochondria allows energy via respiration to be released, and so it gives the sperm the ability to move. And the egg cell, in comparison to the sperm, significantly bigger, a very large cytoplasm, and that acts as an energy store for when fertilization does take place. Now, two specific parts, if we have a look for plants, if we look at leaf cells, their key role is all about photosynthesis. So they have lots and lots of chloroplasts, and the chloroplasts are all about absorbing light. But the plant also does need to absorb carbon dioxide. It's also got to be aware of the amount of water that it's containing. So within the leaf cells, they have very, very specific guard cells. Within that structure of that leaf, these very specific guard cells, and they surround a stomata. Stomata is a pore. And what the guard cells do is they can either become turgid or flaccid. They move water in or out. And by doing that, they change the shape of the stomata. They change the ability for that pore to be open or closed. And so if a plant is needing lots of carbon dioxide at a given time, the guard cells will function in such a way that the guard cells allow the stomata to open to allow carbon dioxide in. A downside to that is, of course, potentially water can then escape. And so if the plant gets to a point where it now needs to control that water loss, it may well close the stomata by increasing the water within those guard cells and reducing that pore. So there's some specialized cells within plants and animals. Now, alongside this, you then need to be aware of how cells are organized. So if we start at the very beginning, we have our cell, which is our basic building block. And what we find is similar cells can group together to form a tissue. Now, you need to be aware of a few specific tissues for the exam. The tissues that will come up, if they do, will be muscular tissues, epithelial tissues, or glandular tissues. Now, I'll talk about all three of these when we come to describe one of the actual systems what we're going to need to know for the exam. But going back, we have our basic building block, our cell, groups of those working together to form tissues, or we can then find groups of tissues working together to form an organ, for example, the stomach. And finally, we can have groups of organs working together to form systems such as the digestive system, an organ system. So let's take a look at a given organ system for our exam, and we're going to look at the digestive system. Well, we have a variety of different cells, for example, epithelial cells, and these epithelial cells can group together to form an epithelial tissue. Now, the tissues within the digestive system, things like the epithelial tissue, which is a covering on the outside and the inside of the stomach. We've got the glandular tissues, and these allow digestive juices to be produced. And we've got muscular tissues, so the actual cells that can move, that we find in the stomach when we're churning up our food. All of these form very specific key roles in this system. And then we have a variety of organs, so the stomach, the intestines, large and small, the liver, the pancreas, all working together to allow digestion and absorption of the nutrients. And so going through some very key aspects to this, we need to be aware of the roles of the glands, such as the pancreas and the salivary glands, producing the digestive juices that we need. Things like the stomach and the small intestine, both allowing the digestion of food. The liver, producing bile, and we'll talk about that later. The small intestine, where the food is absorbed, and the large intestine, to reabsorb water. All of these key parts working together to form a function for an animal of digestion. Now, the other main system that we need to be aware of is then in plants. And this is specifically the role of a leaf cell. The process being photosynthesis. Now, what we'll do, I'll come back to this one very shortly when we start talking a little bit about plants and photosynthesis. So we've got these two key systems for the exam: the digestive system in animals and the leaf, the organs around photosynthesis. Now, just before we get there, final part about cells and processes specifically, some key things we need to be aware of, a couple of phrases. The word diffusion, key aspect to the biology exam. And diffusion is the movement of particles from an area of high concentration to an area of low concentration. Now, specifically, it's the net movement. So particles, on average, are going from high to lower concentration. It doesn't mean a certain particle can't go from the high to low and back again, but on average, we're going from this high concentration to low, this spreading. And this is really important when we start looking at cell membranes. Because a membrane is a type of barrier. Certain things can fit through it, while other things can't. And so we find with the process of diffusion, certain molecules will go from high concentration through a partially permeable membrane into an area of lower concentration, whilst certain other particles could potentially be too big and won't be allowed to pass through. Now, if you're doing the triple exam, you will of course need to be aware of osmosis and active transport, but for the additional processes, diffusion is where we start. Now let's take a look at plants. Like we said a minute ago, the specific organisation within a leaf cell and the tissues therein. Well, if we think the key aspect of a plant and the leaf itself is to enable photosynthesis. But for photosynthesis to occur, we need carbon dioxide, we need water. When we've got some light, which is being absorbed by our chlorophyll within our chloroplasts, we're going to have two products. We're going to have ourselves our glucose and we're going to have some oxygen. Now I'll very quickly balance that just to be 100%, but if you're doing foundation tier, simply the word equation will be absolutely fine. In all likelihood, if you're doing higher tier, I very much doubt you're going to be needing to talk about this specifically, but this is the balanced equation should it come up. So the plant needs to essentially get some carbon dioxide, get some water, and get some light. And then by doing that, it will have produced some sugars, which itself can use for respiration, and obviously we then benefit from when we eat plants. And as a byproduct, there's some oxygen, again used in respiration and also used by us. Now specifically, if we're going into photosynthesis and we're looking at the structures that allow this to happen, here's a very quick schematic of part of a leaf. And there are some key things that we just need to be aware of. First of all, we have lots and lots and lots of palisade cells. And the palisade cells have lots and lots of chloroplasts. The chloroplasts at the very, very top in all of the cells here, allowing for lots and lots of light to be captured. And that is enabling photosynthesis to happen at a steady, good rate. We then have a couple of other things. We've talked previously about this. At the bottom, we have our stomata. Our stomata are our pores, which allow carbon dioxide in, and they do actually allow water and oxygen out. and they're surrounded by guard cells. We then have two other key vessels that we need to be aware of. We have the xylem. Now, the xylem is a way that water is transported through the plant. It's dead cells, so the water is only allowed to flow in one way, and that is up via the transpiration stream, but it will allow the water to get to all parts of the cell. And we also have the phloem. And the phloem are live cells which allow sugars and other key materials for a plant's growth to go in a bidirectional phase, so up and down the plant for where it's required. So our key part to a plant and its adaptations and its structure in the exam will be looking at a leaf, and essentially lots of chloroplasts for photosynthesis, stomata surrounded by guard cells to allow gases in and out, and xylem and phloem which allow water and sugars to be carried. Now, if we go back specifically to photosynthesis and we remember what it is that a plant needs: carbon dioxide, water with some light, and we're going to make glucose and oxygen. Well, a plant has an ability to increase or decrease this rate of photosynthesis. And if we're growing plants, we also artificially have the ability to do that. And this is where we have ourselves this key phrase: limiting factors. There are certain things that we can change or can be changed which will change the growth of a plant. So let's have a look at all three that come up in the exam that we need to be aware of. Well, the very first one is light. If we increase the amount of light, we will find that our plants grow quicker. They grow at an increased rate. However, there be a certain point, as depicted in this graph here, where increasing the amount of light along the x-axis makes absolutely no difference to how quickly this plant will grow. And this is the reason why. Something else is limiting its growth. We've got lots and lots of sunlight, but now either carbon dioxide or water are a limiting factor. And so it doesn't matter how much light we give that plant, without these other two being increased, the plant's growth will reach a maximum. To allow a plant to continue growing at a greater rate, we would need to add either some more water or some more carbon dioxide. And that's the same for our second graph. Slightly different this time. This time we're looking at the carbon dioxide levels. So as we increase the amount of carbon dioxide, we find that the plant itself grows at a greater rate. But again, there's a point. At this point, it doesn't matter how much more carbon dioxide you give the plant, it's not going to grow at any increased rate. And again, this time, there's a limiting factor. But this time, it's not the carbon dioxide that's in plentiful supply. This time, it's either the water or the amount of sunlight that we're limiting. Now, the only slightly different graph that we need to be aware of is one of temperature. And we'll talk about this when we talk about enzymes specifically. But as we increase the temperature, we do increase the growth of the plant. However, if this continues to increase, rather than it leveling off, at this point, we actually find but the rate of photosynthesis dramatically drops. The temperature has gone beyond that of the working range of the enzymes. Worst case scenario, we've denatured the enzymes. So we're going to find that our plant begins to suffer and it potentially then dies. So a limiting factor in photosynthesis will be one of the reactants needed for it. But be aware of temperature, because temperature plays a key role with the ability for enzymes to work. Well, that takes us now into the second key part, where we're looking at organism distribution. And essentially, this is to find out how can we count the number of organisms? How can we find out how many there are in a given area? Well, to do that, we're going to use a quadrat. Quadrat is a 1 by 1 meter square, and it allows us to take a sample of something within. So let's take a look at how we can do this. Let's say, for example, we've got a field of a given area. And on that field are a certain number of red flowers. The question becomes, how many flowers are there? Well, quite simply, we could count them. But therein lies a problem. If the area of this field is vast, simply counting them is going to take a very, very long time. In fact, it might be completely unfeasible. It might not be possible to do that. So what we can do is essentially guesstimate how many plants there are. And this is where the quadrat comes in. And there's a few steps for the exam that we need to be aware of. So first thing we need to be aware of is the area that we are sampling. So we need to find that, and to keep it very simple, in this example, I'm going to have a 10 meter by 10 meter field. So I've got 100 square meters of area. What I can do at this point is, in my mind, grid it. I know that I can have ten by 10 by 10 quadrats would fit into that area, because each quadrat is a 1 by 1 meter square. What I can't do at this point to sample is simply place it where I wish, because my mind might well be guided towards placing it just where the flowers are, or I might be lazy and simply want to place it just in the first bit of the field. So when I know my area and I know how many quadrats I would fit into that given place, I now need to get some random numbers. The more numbers I've got, the more accurate I will be. But the downside, the more time consuming it will be. So for example, I might have 5 random numbers picked. It might tell me to place at quadrat 1, at quadrat 21, at quadrat 22, at quadrat 30, and at quadrat 99. Well, the final step is to then go out and sample. So I place my quadrat in area number 1. And actually in area 1, there is nothing. I then place it at quadrat area 21. And in area 21, there is nothing. But area 22 has something, it has 1. And so on and so forth. So area 30 doesn't have anything. And finally, area 99 doesn't have anything. At that point, I've taken 5 samples, 5 different quadrats. And I've got an average of 0.2 flowers across those 5 quadrats I've sampled. I know that because my total number of flowers was 1, and it was over 5 different quadrats. So for every quadrat, I know there is 0.2 flowers on average in there. Well, if I've got 100 quadrats, my estimate would be 0.2 times my 100 quadrats. So I would be estimating there are 20 flowers. Now, clearly in my example, I've drawn 1, 2, 3, 4, 5, 6, 7. I drew 7 flowers, not the 20 that I've predicted. But it gives me a quick way to guesstimate what there is. And of course, like we said, the more times we sample, the more accurate this figure would be. If I picked 100 random numbers, which eventually would be all 100 places that quadrat could have gone to, I would count in exactly all of them, so I'd know exactly how many flowers there are. If I simply did 1, I'd have the least accurate way. So there's a trade-off between the two. Now, alongside all of that, we have a final piece of apparatus known as a transect. And a transect is essentially allowing us to use quadrats, but along a line. So it shows us the distribution along a given line. We use our line using a tape measure. And again, we sample in just the same way using our quadrat. And this would be a particular use, for example, if we were looking at, say, the distribution across perhaps a small environment. So perhaps that environment, as an example, had some grass, it then had a river, it then had some more grass, and then it had something like a tree. If we wanted to find out the number of fish in this given area, we could place our transect from point 1 all the way to the end. There, if we want to actually sample up the tree, we would continue to go up. We'd still use our random number generator to pick some random places. And it might say that our random quadrats are placed as such. And if we were simply trying to find the number of fish in this area, we should find that the only fish happens to be where these quadrats were placed. In other words, the quadrats where the river happened to be. We shouldn't find any fish in the other places. And so it allows us, using a transect, to find out how an environment changes over a given place. Rather than simply saying there are this many fish in this area, we can actually specifically break that down into given parts. Okay, moving on to enzymes. Now, enzymes come up as part of the cell distribution, cell organization, because we looked at the digestive system in humans earlier. And we just need to be aware of a couple of things with enzymes. Well, first of all, an enzyme is a biological catalyst. It speeds up a reaction. It makes it go quicker, but it doesn't get used up or changed itself. It can be reused. And so we have a huge number of enzymes in our body, and we'll look at some specific ones in a minute. However, the key bit we need to be aware of with all enzymes is that they are living. And so because of that, we need to be really, really, really careful in how we actually control them. Specifically, they can easily be denatured. And denatured means they break, they no longer function. And so two key aspects can affect enzymes and how they get denatured, how they break. The first is temperature. Enzymes have a given temperature at which they work best. Now, in humans, that temperature is 37 degrees. Anything above that, they're in danger of no longer functioning well. And if they go too far, they could break. Not all enzymes work at 37 degrees, but the vast majority of human ones do, and that is their optimum. But different organisms will work at different temperatures. Sorry, different enzymes will work at different temperatures. They also have very specific pHs that they work at. So enzymes in the stomach will work very well at around pH 1 or 2. But other enzymes would find that they would denature at that given pH. So we just need to be aware of key things that can cause enzymes to denature. So living organisms produce these very specific proteins known as enzymes, and they are very, very, very specific ways in which they can function based upon temperature and pH. Having said that, I just want to very, very quickly correct myself, because I think I said right at the very start of this that enzymes are living. What I was meant to say, I do apologise, is that enzymes are produced by living organisms. They themselves are not living. They're complex proteins that are folded in given shapes, but produced by living organisms. Now, for the exam, we need to be aware of a couple of very specific enzymes. And these enzymes allow all sorts of different functions in the body. We'll go relatively quickly for this, but some enzymes are involved with respiration, and we just need to be aware of the equation for respiration. It's the opposite of photosynthesis. It's taking glucose and oxygen, and it's producing carbon dioxide, water, and we're also getting some energy. So these are the key parts for respiration that comes up, specifically aerobic respiration. If in the exam you get asked about anaerobic respiration, well, that is respiration without the oxygen. And so actually, rather than producing carbon dioxide and water, we actually get lactic acid. We do get some energy, significantly less than aerobic respiration, but it's this buildup of lactic acid, which means we have a certain time where we can anaerobically respire before we have to stop and go back to aerobic respiration. This lactic acid is very difficult to be processed by the body, and so we have at the end of vigorous exercise an oxygen debt where we breathe in deeply to get as much oxygen back in to break down this lactic acid back into carbon dioxide so we can remove. With the specific enzymes that we need to know for the exam, there are 3 key ones that our body makes, and then 1 in industry. The first is amylase. It's also known as carbohydrates. Do apologise for the pen. Amylase takes starch and it breaks it into simple sugars. The second that we need to be aware of is protease. And protease takes protein and breaks it into amino acids. Finally, we've got lipase, and that takes a lipid, a fat, and breaks it into glycerol and a fatty acid. I did say earlier that there was a product called bile, which is integral in the digestive system. And bile is produced in the liver, released into the small intestine. It not only neutralizes the acid, but it also helps at emulsifying fat, breaking down fat globules into smaller globules, allowing lipase to function. Now, there is one final enzyme that we need to be aware of. It's not made in the body, but enzymes are used in industry, and it's known as isomerase. And isomerase allows glucose to be broken into fructose. So it's found in sugar-reduced drinks, things like Lucozade Light. And it works because fructose is a lot sweeter. Because it's a lot sweeter, you need less of it. And so it's a lighter sugared drink. We also have enzymes that are used in detergents. Baby food has protease in it to break down proteins because babies don't quite have the function yet to break down proteins themselves. Finally, enzymes are used highly in industry. They're very useful at speeding up reactions, so that lowers the cost, makes it worthwhile for the companies. But they do have their downsides. Obviously, as we said previously, they can be denatured, so we need to look after them. They can be quite expensive. And the last thing you want is your enzymes to actually be used in industry to then end up in the product. If you were using isomerase to break glucose into fructose, you don't want that isomerase to move into your product. Now, they're often found on special beads where the liquid, in this case the glucose, will be poured through, being broken down as it does and coming out as fructose, and the beads would remain there. But these are relatively expensive processes to set up. Now, moving to part 2, we're going to look at DNA, and we're going to talk about Mendel, genetic chromosomes, crosses, and we're going to end on a little bit of evolution, fossils, and speciation. So first off, chromosomes. Well, chromosomes are long strands of DNA found within the nucleus, in animal cells, and the DNA itself is our genetic material. It's what we inherit. It is what makes us us. And so the DNA is unique, exception obviously being if you're an identical twin. But it allows instructions, it allows proteins to be produced, and then those proteins build up and up and up and work together to form more complicated organisms. Now, specifically, the DNA, which is found within chromosomes, can actually be read as small parts. These parts are known as genes. Genes are small sections of DNA, and they code for something. They code for a very specific protein. So DNA, think of it like the alphabet. The genes are small words. They're all found in chromosomes, the book, and all the books, all 23 pairs in a human, are found in the nucleus of an adult cell. Now, specifically, you need to know 2 ways in which cells can replicate. If If you're doing foundation, you need to know mitosis. If you're doing high, you need to know mitosis and meiosis. If you're doing foundation, you do need to know the word meiosis and what it refers to, but you don't need to know the specifics. So starting for everyone, first of all, we have ourselves 23 pairs of chromosomes. One pair of sex cells. With every human, 23 pairs of chromosomes. And mitosis is essentially a way to replicate one parent cell into two cells. So for example, as we age, as we knock some skin off, as our epithelial cells within our stomach and small intestine begin to break away, we need to replace them. And this is the process by which this happens. First of all, our parent cell, a normal body cell, has the DNA which spreads out into long strands. At this point, we find that a signal will be taking place in the cell, and that signal is telling the cell it's time to divide. The first thing it does is it duplicates its DNA. If we make up a percentage here for simplicity's sake, it's a normal parent cell, 100% cell, one of our normal body cells, and we get a duplication. We get a duplication to 200%. We then get a very specific lining up of the chromosomes. And we get these special spindles that come down and pull the arms apart. The chromosome ends go in opposite directions. We then get a new membrane falling and the cytoplasm dividing. It's divided completely equally. So we now have two cells, both containing 100% of the same DNA. We started at 100%, we duplicated up, we got a split across the middle, so each chromosome was pulled apart perfectly in half to allow us to have two cells at the end of the process, both containing 100% DNA. And that's mitosis. It's taking one normal cell and duplicating it into two. Now, meiosis is slightly different because meiosis is how we produce gametes, sex cells. And rather than having 23 pairs or 46 chromosomes, they have 23 chromosomes. They have half the amount of DNA. And furthermore, the DNA that they contain is different between each gamete. It's a random selection of DNA, which is why siblings from biological parents, which are both the same, are not themselves identical. And so this is how meiosis occurs. Well, as in the case of mitosis, we have a trigger for a normal adult cell to divide, a normal adult body cell. And we get a duplication of the DNA. And like we had with our mitosis example, after this duplication, we get a division. The pairs line up in the center and are pulled apart. However, they're not lined up perfectly. After the duplication, we don't find that the left-hand side and the right-hand side are identical. We have had our first division, but we've had a little bit of randomization. We've got some red cells, red chromosomes on the left-hand side, and some blue on the right. Well, at this point, we have, after our first division, another pulling apart. And so now we find that we only have one chromosome pulled apart. We've got this second division. And at the second division, again, our chromosomes have lined up just like they did in mitosis, but they're pulled apart again. And we actually find that in this second division, we get another randomization. And what do we end with? We end with 4 daughter cells, each containing 50% of the DNA. We had a normal cell at 100%, but it was doubled up. We had a division, first of all, going back down to 2 cells with 100% DNA. But we then get a further division where we have 4 cells, each with half the DNA. And the key aspect, we've had 2 points where we have had a division that isn't identical. We've had some randomization. And so all 4 of these gametes, although they started with exactly the same initial DNA, will be different from one another. And when that is merged with a different set of gametes from the partner, for example, if these are 4 different sperm cells, from the mum, there would be 4 different egg cells. We get quite a lot of randomization. And so although siblings are 50% their mum and 50% their dad biologically, the 50% of dad that one sibling gets and the 50% of mum that another sibling gets can be different. And in fact, statistically, are highly likely to be different, which is why the vast majority of people who are close relations have similarities, but also have differences because they've taken different parts from mum or dad. Finally, with DNA, we need to be aware of stem cells. Now, stem cells have the ability to turn into any type of cell. They are undifferentiated. They can become anything. And so this is a very, very powerful thing to have. If we take embryonic stem cells, well, potentially these embryonic stem cells, these cells that haven't become anything yet, could go off and become a nerve. They could go off and become muscle. They could go off and become potentially a whole organ. So having an undifferentiated stem cell, having a cell that hasn't specialized into anything, is very powerful. So for example, if there was an individual who suffered some kind of trauma, perhaps they had a broken back that severed a nerve cell, there's a potential with stem cell technology to take embryonic stem cells that haven't differentiated, placed into the broken part of the spinal cord, and maybe in a few years to come, they could re-specialize into nerve cells and bridge gaps and allow paralysis to be a thing of the past. So they're very powerful things. We're not there yet, and we need lots of research to get us there. And of course, that in itself raises lots of questions. Many people are against the use of stem cell research. If we're talking about embryonic stem cells, Well, they tend to come from fertility clinics. Once with the right specifics, that embryonic sperm cell could become a living being. And there's an argument, should you be taking that embryonic stem cell and using it for medical research? It doesn't have a voice of its own. So there's arguments that you need to be aware of for should or should they not be used. The counter to that, if they were fertility clinic embryos that were not going to be used medically, they would be destroyed. So if they're going to be destroyed, why not get some use out of them? Again, no right or wrong here, but you need to be aware of the arguments with it. Okay, we're now going to get on to Mendel and genetics. Mendel, the father of genetics. With genetic crosses, we need to be able to work out percentages of likelihood. So some key aspects that we need to be aware of, and we'll give some examples as we talk you through this. The first thing we need to be aware of is this phrase, an allele. Now, an allele is a gene with two different variants. And we call these two different variants, based upon this allele, either having dominance or recessiveness. For example, if we take someone's eye colour, it could, in this example, be brown or blue. The pigment eye colour is an allele. In this simplified version, there's one of two options. In the real world, there are many, but for our example, they could either be brown or blue. The pigment will always form, but one of two colours will form within. Well, in this case, we have a dominant allele, brown, and a recessive allele, blue. The dominance is shown by a capital, the recessive a small. Furthermore, we just need to be aware of some phrases that they might say in the exam. We have ourselves homozygous dominant. Homozygous meaning the same. And if it's dominant, they would both be capital letters. We could have ourselves homozygous recessive. Homozygous the same, but recessive, so small letters. Or we could have heterozygous, different. In other words, one of each. Now, based upon all of that, we then have ourselves the ability to predict outcomes. Let's look at the chances of having a boy or a girl. We all know that to be 50-50. But why is that? Well, this is where a Punnett square comes in. We need to take the two parents and find out what are the chances of the offspring. So let's do that. We make ourselves a very simplified Punnett square. There's a few other ways that we can do this, but this is very much the simplest way. We then take the parents. So here is the mum, XX, and along the bottom, going vertically, the dad, XY. What are the chances of a boy or a girl? Well, we simply work down and across. So X going down, X going down, X going down, X going down. Then we work across. X going across, X going across, Y going across, Y going across. We have 4 potential outcomes, of which 2 are for boys and 2 are for girls. It's 50-50. Let's go through a few more examples. Say we had ourselves an individual with some given disorders. I'm going to take, for example, here, cystic fibrosis. Cystic fibrosis is on the recessive gene. So I know that I need 2 recessive genes to suffer from it. Let's take some examples. Say my wife and I are both, in all sense and purpose, normal. We do not have cystic fibrosis. That does not mean we do not carry it. And let's have a look how. Let's take someone that doesn't suffer from cystic fibrosis, the dominant allele, to be a capital F. But a sufferer is a small f. Well, there's a few outcomes here. If we do our Punnett square, let's have a look at what we can get. Well, what if we're both genetically dominant? Well, we know the outcome there. There, for example, is me. There is my wife. Going across and down, all 4 outcomes would be children without cystic fibrosis. Nor do any of them carry it. Absolutely all of them would have no issue. But actually, there's a couple of different scenarios. What if my wife was normal and had no cystic fibrosis, but I carried it? I don't suffer from it. The dominant allele is winning, so I feel fine. But I'm a carrier of the recessive allele, the allele that can cause cystic fibrosis. Well, in this case, all of my kids would still be fine. 2 of the 4, so a 50% chance, would be fine and wouldn't carry it. But the other 50% would be fine, but would be carriers like I was. But what happens if my wife and I both suffer from it, or we are both carriers from it? Well, again, let's take a look. If my wife was fine and I were fine, but we were both carriers, now what we would find is the following. Here, I've got a heterozygous, sorry, I do apologise. Here I've got a homozygous dominant offspring. This child is fine and they don't carry it. These 2 children are fine as well, but they're carriers of the disease. Unfortunately, this percentage, 25%, 1 in 4, or the ratio 3 to 1, this child would suffer. And of course, that would go up should my wife or I actually have the disease. If my wife had the disease and I was a carrier of it, we would find the following. We would find now that actually my chances have suddenly changed. I've got 2 children who suffer from it, a 50% suffering rate, and I've got 2 children that are fine but are carriers. So on the recessive, we can work out the percentages based on homozygous, heterozygous, and dominance or recessive. Now, if we take the second example that we need to be aware of, polydactyly, polydactyly is on the dominant. Polydactyly is extra fingers or toes. And it's on the dominant gene, the dominant allele. So I'm going to use big P for polydactyly and little p for not having polydactyly. And again, we can look at it as we did previously. The only way you can not have polydactyly is to be homozygous recessive. But you could be having polydactyly by being homozygous dominant or heterozygous, because this time it's on the dominant. And so if you took one individual who was absolutely a non-sufferer of polydactyly, but they were to have children with a carrier of polydactyly, then again, across, and down, we would find the following. We would have ourselves, in this case, a 50% chance of having a child with polydactyly and a 50% chance with not. But if we had ourselves an individual who was homozygous with polydactyly, who had a child with a person without polydactyly, suddenly our percentages become 100%. So in other words, genetic diseases on the dominant alleles are far more likely to be passed on than those on the recessive. Finally, just to be aware of the types of genetic diagram that the examiners will like to ask, a Punnett square, in my opinion, is always the easiest way to do it. But if they give you the non-Punnett square type of question, there's a couple of simple ways to look through it. We start with the genes that our parents would contain, of which there will be two different alleles in these cases. And then when we've got those two, whatever they happen to be, they will go on to lead into 4 outcomes. And the way to remember it, on the left-hand side, we go down to the left and down to the left on the other side. On the right-hand side, we go down and then down to the right. So left is down, and then we have ourselves, sorry, I've drawn that there. Sorry, let me start again on that one. Right, sorry, let me try and be a little bit clearer. So on the left-hand side, we go down, and then we go down to the left. The right-hand part, we go down, and then we go over to the right. And then on the right-hand side, we go left and left, and then on the furthest right, we go right and right. It's a bit of a complicated way to do it, but they may well ask you in the exam to fill in the blanks at the bottom. Finally then, we just need to be aware of fossils, speciation and extinction. Well, the key bit with fossils, the oldest fossils are found at the lowest part when we dig, and that's because they were buried the furthest time ago. What are fossils? Well, they're remains of plants and animals. And there's three different types of fossilization that can occur. We can get the gradual replacement by minerals. So teeth and bones, which don't decay very easily, are buried, and eventually they're replaced by minerals to form a rock-like substance of how the animal once looked. Or they could be casts, impressions, so an animal stood on something, brushed up against something. And finally, there could be a preservation, such as animals found within amber. Now, fossils give us a huge amount of insight into what life was like, maybe how life hasn't changed over millions of years and how it has. And it gives us an idea of what is living at certain geological points in time. But the key bit with all fossils is that they are very rare. The process of fossilization is one that needs some very specific circumstances to occur. So the animal itself needs to have not been eaten straight away. It needs to be buried relatively quickly. And then most importantly, we need to come along and find it. So it's a rarity. But it offers a good insight into how life on Earth began. Of course, the earliest life forms were soft-bodied, so we don't find fossil records of them. Furthermore, 4.5 billion years ago when the Earth began, and 3.5 billion years ago or so when life began, well, we weren't around. So it's very, very difficult to be sure. But the fossils give us a very good clue as to how life began and how it has changed over time. Finally, extinction and speciation. Well, extinction is if an organism or a species goes extinct. In that case, there are no more examples of that individual. So, for example, a woolly mammoth, their species go extinct. There are no more woolly mammoths worldwide. What can cause it? Well, a variety of different aspects. Number one, there could be a new predator, which simply eats them all. There could be a new disease, which they can't acclimatise to. There could be new species, which can outcompete them. So perhaps they're outcompeted for food, for shelter, for space. Or there could be some kind of climate change, some kind of new environmental change. That could be a catastrophic disaster. It could be like a meteorite, which we think was what caused the wiping out of the dinosaurs. Or it could sadly be human activity and digging up for raw materials and reducing the natural environment for certain organisms, such as the snow leopard. Well, finally, to end, speciation. We just need to be aware of how new species come about based on natural selection via evolution, the driving force behind it, whereby you have individuals that show some variation. Life is tough, so only the best adapted survive. If they're more likely to survive, they're more likely to pass on their genes because they can reproduce. And so those genes get passed down to the next generation and so forth. But the mechanism of a new species to actually come through and evolve via natural selection can begin relatively simply. If we have a common population, such as that shown in figure 1, if something separates them, for example, a mountain range, or some kind of reason why these two communities can no longer interact, over millions of years, maybe less, if the environments are slightly different, we might find that the species adapt differently, pass on different genes, and over time become different. If over a given amount of time, those changes are so extreme that if the new populations were to be reintroduced, they could no longer successfully breed and produce viable offspring which themselves can then breed, we would deem that a new species. So speciation is the separation of a common population where they cannot get back to one another. Over time, with different environments, different changes may occur. And if that population were allowed to then be reintroduced, they might have changed enough where they subsequently are unable to breed with one another. Well, that brings us to a close to that Hangout. I do apologise for the initial part of the presentation, and I'm sorry that the pen wasn't particularly clear. Hopefully we'll sort those teething problems out for next time. I wish you every success in the exam, and as previously, we are online for any specific questions that you wish to place. Thank you very much. Good evening.